The ones I've graded so far have looked good. I expect I should have them all graded and released to you before we meet again on Thursday. Oh, cool. All right. We have some people who are having a viewing party. I like it. But yeah, so we should have, I should have those released to you before we meet next time, or at least very close. Very close. I graded all of the manuscript reviews for the first one. What you did on the first one, there were a few people I maybe made comments to, but it all had to do with like keeping the heading in. Like you can leave the headings in because it makes it easier to sort the grade or to go through and evaluate it. Otherwise, everybody did excellent job. So whatever you did for the first one, keep doing it for all the rest, and you'll get all of that credit. They were very good. Awesome. So I got an email, and I replied to the person emailing, but also I thought I would just state it out loud too. So exam 2, I believe, says that it'll open. Hopefully, it's next Friday. That it'll open next Friday, and then it'll be due on Sunday. And they asked, will that be what it is, or will it change closer to what we did for exam 1? It will probably change closer to what we did for exam 1. And I set up those due dates so that they're approximate, so that in your when you go on Canvas in your syllabus, it will go ahead and populate when things are due, so you kind of have an idea. Selfishly, it works better for me to have it posted, to have it available starting Saturday morning, and then I'll give you until Monday to submit it. Now, I think, and I did not look, so I could be wrong, but I'm pretty sure that there is there's one exam that is due in the middle of the week, and that was on purpose, and it was to keep the exams from getting clustered together where they were really frequent. As we get closer to that, whatever exam that is, and I think it was 4, but it could have been 3. I don't know. As we get closer to it, we can all discuss how we'd like to see it done. I just tried to give as much space between each exam as possible. So yeah, when you look at the due dates on Canvas for the exams, that's to get you thinking this is the approximate time. Cool. All right. I don't think I have anything else. So what we'll do, as soon as I can find the record button, is we will finish out chapter 5, and we will start, maybe finish, see how it goes, chapter 12, which is our cytokines.
This meeting is being recorded.
Hooray. Good. All right. So where we left off, we were covering our different isotypes of antibody. And so I will ask it, feel free to send it in the chat or just think about it, but ask yourself, what does it mean to be an isotype of an antibody, right? What is that isotype based on? And what you have to do then is think back to antibody structure, right? And to say, well, how is an antibody built? It has heavy and light chains. There are 2 light chains in an antibody molecule, each of which has an Ig fold domain, right? And that's what are these little lumps. Often you'll see them drawn this way. We'll look at a couple of other images from a different book that draw them a slightly different way. But these little domains, which are a particular way that the amino acid sequence gets folded and clustered together, are unique to a superfamily of molecules called Ig superfamilies. We have a slide or two at the end to describe those. And so what we were doing is we were going through our isotypes, and isotypes are based on these Ig fold domains on the heavy chain. And it turns out there are 5 genes that encode for those, and that gives us our 5 isotypes. A few, it's principally IgG, which has 4 subtypes, and I are subclasses, and then IgA has two subclasses, and those are ones where they're still genetically they are still very similar to each other, but they are not very similar to anything else or to any of the other isotypes. And so as you're going through these and you think, oh, well, what does it mean to be IgA? It means it has a specific genetic sequence that's encoded for the constant regions of the heavy chain. Now, this is a good place to remind you that you have on an antibody two locations where antigens will be, or epitopes will be bound. And what those are based on are these variable regions. So these are an Ig fold domain that has really high variability, and that's what gives you the diversity of a repertoire of possible epitopes that can be bound, right? And that's in part due to each of these variable domains has these CDR regions, which are hypervariable. So you see a lot of mutations, and the types of mutations are not different genes, but instead they are the way that the one gene gets rearranged, mixed and matched. All right, all that said. Oh, somebody asked allotypes and idiotypes. Yeah, so the simple version is an allotype. So you have three different variants, right? You have isotypes. Here, I'll click back. You have isotypes, which are based on which heavy chain is being expressed. You have allotypes, and allotypes are where you could have two IgG molecules, but you have a very small mutation that is in that heavy chain. And really, from a practical standpoint, these only play a significant. These only have significant consequence when you're talking about inheritance, right? So we have here two antibodies, both of which are IgG, but one of them has a mutation that can be tracked because it follows Mendelian inheritance, right? Is there any reason that this mutation could affect antigen binding? No, right? Because antigen binding is occurring in the FAB region. You could make an argument that you could have a mutation in some of these constant chains, and it could affect FC function, right? That antibody might not bind its receptor appropriately. So know that, and use that as a way to reinforce what the FC portion does and is. So that's an allotype, right? There's still it's still the same isotype, but it has a mutation. Idiotype is the one that's a little bit more abstract, and this is the idea that very simply they are going to be antibodies that are built that would respond to an antibody itself. And depending on which part of, because remember, if you get an antibody from somewhere else, that makes a lot of sense. Oh yes, of course, it has to generate an immune response, right? We got an antibody from a pig. Well, we are not pigs, so our body will see those portions as foreign. However, as you produce an antibody response, you will also eventually develop an antibody response to your own antigens, but it's going to be concentrated on these variable regions. And why does that make sense? Because those variable regions could be things that are not recognized by the immune system, right? There's so much variability. And so idiotypic antibodies produce a response that is termed an anti-idiotype antibody. And that's a way to help modulate the antibody response. It can give you interesting things. If the idiotypic antibody binds to a portion that's not involved with antigen binding, the antigen will still bind. However, if it binds into the cleft or this little pocket or the specific epitopic determinant, then it can interfere with it. And so isotypes are the one we care most about, right? Based on heavy chain. Allotypes are the same heavy chain that has a minor mutation, won't affect antibody binding. And then idiotypes are antibodies that recognize other antibodies that are developed because as you produce an immune response, those variable regions are somewhat foreign. Cool. Yeah, hopefully that helps. And that idiotype idea, like we could spend more time going into it. Your text doesn't, which I'm glad for. So kind of, if you know the terms and know the general scheme, you should be okay. All right. So with that in mind, right, a good refresher of the variants. IgA is an antibody that, for practical purposes, you are going to find it in a bivalent form, which means it's going to be two immunoglobulin molecules that are attached together. Anytime you see an immunoglobulin attached to another one, if it's forming a dimer or these pentamers, it's going to be through something called the J chain, and that's what helps form covalent linkages between them. Now, you can find IgA in a monomeric form where it's just your traditional little IgA. You find this in the serum, and for our purposes, it has no known or well-characterized biological function. Now, the dimer form, and where we speak about IgA most often, is in the context of external secretions. You see IgA in saliva and mucus and sweat and the secretions of the stomach and the small intestine, tears, and importantly, the colostrum of breast milk. So you're going to find that anything that's being secreted to an outside surface, you're very likely going to find IgA present. Again, dimer in this case, joined by the J chain, and because you can find it in the colostrum of the breast milk, it's very likely has a role in giving passive immunity to a neonate, right, a breastfed infant. Now, we can look at our molecular weights again. The monomeric form is 165 kD, whereas the dimer, in addition to having some other things that we'll talk about, the secretory component and the J chain, it's closer to 400 K. And kind of, help yourself use that table in the text to kind of rank where those go. IgM is your biggest with IgA. Why are those two the top? Because they are dimers or pentamers. And we have two subclasses, IgA 1 and 2, although when you look at the proportion of IgA in secretions, it's 93 or so percent IgA 1. So when we speak of IgA, we're generally speaking of IgA 1 because it's so prevalent. Now, it has a fairly short half-life at 5.5 days. And if you think back and compare this to IgG, which for the most part has very long half-lives, what gives? Like, why does IgA have it? It's thought that in the serum, its IgA is so short because since it's a monomer, it is unable to bind the secretory component, which makes it less stable. So it's quicker to be recycled or eliminated. And again, no clear biological function. Now, what's termed secretory IgA is always a dimer. You're going to find it in these fluids associated with the external environment. So you see these in the barriers. And it turns out IgA plays a vital barrier defense role. Some immunology texts have a whole chapter about mucosal or barrier immunity. And a large part of that from an adaptive response is IgA. Plasma cells are who, again, secrete our antibodies, and reminder that these are just B cells that have seen antigen and have clonally expanded and differentiated into robust antibody producing factories. Now, to get this secretory IgA to these surfaces, you're going to find these plasma cells, instead of hiding out in the lymph nodes where you see some, or the bone marrow, plasma cells producing IgA tend to make themselves very close to the surface epithelium, and they exist they migrate to a place called the lamina propria. Now, once these plasma cells in this lamina propria area release their antibody, what happens is, and it comes out in a dimer, it actually binds to, again, our poly Ig receptor. Poly meaning, kind of fairly ubiquitous, but on the epithelial surface, it'll actually bind these IgAs, and it will draw it across, or it will bring it into the epithelial cell, and it'll start to chew it up. And in the process of chewing up this poly Ig receptor, it creates a dimeric IgA that has this secretory component, which stimulates it to be released. Because of that J chain, and because it's a pentamer, IgM can also be secreted in small amounts. So poly Ig can grab a little bit of that. Now, what it's thought is, if you take the secretory component off IgA, it is more sensitive to all of, think back to chapter 2, right? All of these enzymes and chemical barriers that are related, or chemical mechanisms of innate immunity, mucus and saliva and tears, those are inhospitable places, and that's by design. And so to protect this dimeric IgA, if it has the secretory component, that helps to minimize how susceptible to enzymatic activity it is, which means it lasts longer in the serum, or in these external fluids. So you have a plasma cell, we would call this the lamina propria, right up against the basement membrane. It secretes its dimeric IgA, which is going to bind to poly-Ig receptor. It grabs it, brings it in, and through a process of degradation, it ends up cleaving away the poly-Ig receptor, leaving a secretory component, which helps protect and ensure that that antibody persists in its dimeric form. As you would expect, if it's getting secreted, you're going to see that lots of IgA is in the mucosa. It seems like it plays a big role in neutralization. So what it does, one of the things it can do very well is to help prevent or reduce attachment of pathogens to the epithelium. That's one of the first things a pathogen must do to bypass the epithelial barrier. What that does is it limits the entry. What's interesting is that it does have bactericidal activity, which means it can cause the death of a bacterium, but it seems to be more specific to gram-negative, and it seems to only work in the presence of lysozyme. And we won't get into the mechanisms for that. So instead, one thing to remember is, yes, it can cause bacterial lysis because of lysozyme, but it does not do so by activating complement, right? IgM activates complement, and IgG can activate complement. Because it's a barrier, you would expect, because this is a barrier defense, you'd also expect it to be pretty good at limiting viruses. And it does. And it's especially good because it's a dimer. It has longer reach for those ultimately 4 antigen binding sites to reach. It's a very good agglutinating antibody, which also renders a viral pathogen, in this case, unable to bind the things it needs to. IgD is honestly an antibody that we would talk about more in a different context. And so we're just going to have a brief word, and that is IgD is very rare in the serum because development of IgD secreting plasma cells is very rare. IgD is one reason there aren't a lot of plasma cells, but what little IgD might get secreted into the serum is fairly quickly degraded because it has a really long hinge region here. And that hinge region means that it can be clipped and it kind of dissociates. When a B cell gets activated, they rapidly downregulate the delta chain, which again, delta chain are those heavy chain constant regions related to IgD. And as a part of the genetics of the B cell receptor and of antibodies, that it just is cleaved out more quickly. Now, there is IgD expressed though. It gets expressed on the membrane, so you don't find much in the serum, but you can find it on B cells. What's interesting is that it can be co-expressed with IgM, so you can find B cells that not only express a B cell receptor with IgM, but they can express a B cell receptor in tandem with IgD. Again, it's going to be an antigen-specific receptor, and what's interesting is you only find this membrane IgD expression in the context of a mature B cell. So one of the things from a research standpoint, or maybe clinical, you could use expression of IgD to say, do we have mature or immature B cells? Now, there is some evidence that membrane IgD plays a role in helping to get rid of these B cells that are binding host or self antigens during development, right? And so it binds those and it helps get rid of them to clear them out. So we'll finish with IgE. Everybody usually has a working understanding of IgE. It has a molecular weight of 200 kD. One of the reasons this is heavier than our other single monomeric antibody is it actually contains a 4th constant domain on its heavy chain, right? Typically it's on a heavy chain variable and then 3 constants, IgE and IgM, but IgE has an extra one. It has the shortest half-life of only about 2 days, and it's incredibly rare in serum, and that's possibly beneficial because we associate IgE more so with allergic responses and hypersensitivity. Now, what's interesting is because of this extra constant domain on its heavy chain, it has incredibly high affinity at binding with Fc epsilon receptors. And these are expressed principally on mast cells, basophils, and eosinophils for our purposes. And so what it can do is it can actually create this response where you have very little made, and what is made binds very well to using the Fc portion to cells that express that Fc receptor. And so what you get are IgE labeled granulocytes. These are neutrophils that are moving around that have IgE decorated all over it. What happens is, in this case, if that now membrane bound IgE binds its antigen, it can induce degranulation. And what degranulation means in the context of, say, basophils, mast cells, or eosinophils is they spill all their guts and they will induce an immune response. When IgE is bound to an Fc receptor, when it's decorating a membrane in this case, its half-life increases from a handful of days to weeks or months. So it becomes incredibly long-lasting. And this is why, if you think of a type 1 allergic response or type 1 hypersensitivity, right, that threat is always there. And that's because the little bit of IgE that gets made preserves very well because it's decorating all these cells that can induce an inflammatory response. Now, we think of IgE as being bad in the context that it drives allergic reactions, or at least particular types of hypersensitivities. But it's also an important aspect because what IgE promotes is parasitic responses, anti-parasitic responses. If you take individuals who have been immunized with an Ascaris antigen, which an Ascaris is this, can be very grotesque worm that's a parasite, so it's a type of roundworm. And if you immunize people with these, what happens is they start to produce lots of eosinophils and they start to make lots of IgE. And what those two things can do then is help to have that anti-parasitic response. What's interesting is that we think of IgE as driving these robust inflammatory reactions, but they do not do so by activating complement. Their action is principally the result of causing the degranulation of mast cells, basophils, eosinophils. And in doing that degranulation of those types of granulocytes, they release histamines, heparins, and leukotrienes. And those are potent inflammatories, right? They're vasodilators, and they're going to cause those symptoms of inflammation. And so if you think about how you would treat somebody who has, say, an allergic reaction to something, what do you typically do? You might give them antihistamines, right? So you're trying to counteract the activity of IgE inducing the degranulation of some of your inflammatory cells. Now, when you look at the antibodies, you can talk about their kinetics. Really, this maps very well directly on top of our primary versus secondary immune response. So here, we're just going to bias it towards antibodies. Primary response, again, is the first antigen exposure. We can divide that up into four phases, which are obvious, right? You're going to have the period where everybody's getting activated. You need antigen presenting cells to grab it, B cells to get activated, plasma cells to become formed so that we can produce it. And so you'll have this lag phase of 1 to 2 weeks where you don't have any detectable antibody. And then you'll have periods where it's increasing in the exponential, where it's balanced with the steady phase, and then where it's starting to decline. This is important because this is when the immune response is contracting, which means it's turning off because the threat is being eliminated. Now, in the primary response, what you'll see first is the production of IgM. When we talk about B cell activation, we will reinforce that as soon as the B cell gets activated, it starts making IgM right away to kind of say, hey, let's get this pathogen. But over time, the more it starts to expand, the more it'll shift and do a series of things to start to produce, or the antibody response starts to lead to IgG, which is more effective than IgM. Now, secondary response is when it sees that antigen again. And here you're looking at an immune response from memory cells that were formed as a result of the first antigen exposure, which means shorter lag phase, a much higher magnitude of antibody production. And it's going to be principally IgG driven. Like most of the secondary immune response you'll see are going to be IgG antibodies. And I should add that here. Now, the magnitude of that memory response and how fast it comes and how much IgG, and honestly, how much or what specific contribution of the repertoires you'll see really are based on many factors, such as the person's genetics, just the environment in which that antigen was seen, and even the antigen itself. Some antigens make better immune memories than others. And we can see that again, right? Primary response, it takes us 10 to 14 days. We see IgM first, and then IgG replaces it. And then upon the second immune response, we'll still see IgM produced, right? Those memory B cells are still going to get activated and produce IgM first, but then they do so much more quickly, and they have a shorter time to where they'll start to produce more IgGs. Now, in the secondary response, this is where you start to see that class switching. This is why you start to make much more IgG. And this is a result of class switching. Class switching is beneficial because if we're just comparing IgM to IgG lasts longer, right? It's smaller, so it's capable of getting into perhaps smaller places, but it takes more time to get produced. And I borrowed this from another text that we may or may not cover in an abridged context, but it shows how you go from an IgM expressing B cell, and you're going to have this class switching response, which actually clips out pieces of the DNA in that B cell, which means that it can no longer express, for example, IgM, D, or this particular version of IgG. And instead, it class switches, it rearranges, it excises out the other heavy chains that it's not going to express. And now you have an IgG expressing B cell. And all that's happening as a result of antigenic stimulation and activity in the secondary lymphoid tissues. Now, depending on the immunogen, you may not have as much IgG, and instead you may, or IgG, you may have, it may drive an IgE or an IgA response. And really the types of class switching you get are all based on the T cell help, right? This is why in the adaptive response, humoral immunity, the production of antibodies is influenced by what happens in the cell-mediated response. Now, when you have this class switching, you are still binding the same epitope, right? That's not going to change. But instead, you are just rearranging and expressing different heavy chains that have the same epitope. And so it allows you to fine-tune your immune response without sacrificing binding that same antigen. And again, this occurs in germinal centers, which we introduced in the lymph node and spleen, but these are areas of high B cell activity as a result of antigen binding. Now, the other thing, in addition to class switching, and I think this came up when somebody was studying for an exam and we chatted about it, you also are going to see in the secondary response better binding of the antigen that did the activating. And this is through a process called affinity maturation. And what this does is you get antibodies produced that still recognize the same antigen and the same epitope, but the weak forces that interact are much better, which gives you more affinity. And this is the result of somatic hypermutation, because we have these CDR regions that are unstable. Well, what happens in the germinal center is that you have these activated B cells, and they get in the germinal center and they start to rearrange their antibody binding sites. And then they test it on the antigen that did the activating. And if it's not a better binding, they get dumped and destroyed, or they'll recirculate and they'll undergo more somatic hypermutation. And so what's really interesting is that not only do you start to make antibody isotypes that have better functions, you also start making more antibodies that are tighter binding. All of that together means a much better antibody response. So a couple of things to finish on is the idea, we've talked about these antibodies, and we're going to see additional molecules. We already talked about some, and these are all going to belong to something called the immunoglobulin superfamily. And this is what these look like, right? We've just looked at them as little lumps, but really what they are these common structural elements called Ig folds. And these are regions that are about 110 amino acids that are actually formed because at the beginning and the end of this little region, you have an intrachain disulfide bond, and it creates this globular structure. It creates this little lump. And in an antibody, for example, the heavy chain has 3 or 4 of these, or excuse me, 4 or 5, and the light chain has 2. And one of the interesting things is that these are so redundant. They can be very homologous between all of these that have these Ig fold domains, is that it speaks to 1, evolutionary lineage, and 2, importance. The body typically doesn't, or cells don't remove something that plays a vital role. Now, this is the largest number of members of the superfamily. It's one of your bigger molecular superfamilies, and one of the requirements is they all contain at least 1 immunoglobulin fold. And so what I've done here is I've just listed a couple of examples that are particularly important to things we maybe have already introduced, but especially things that we will talk about more as we move forward in the different chapters. So we have antibodies, or our immunoglobulins. Remember, we have them secreted as antibodies or membrane bound as B cell receptors. The parts that make them Ig superfamily here are the heavy and light chains. So recall back from earlier in this lecture. We have Ig alpha / beta, or Ig alpha, Ig beta. You will always see these 2 pieces go together. And what these are the signal transducer for B cell receptors. So when we talk about B cell activation in chapter 10, 12, 10 maybe, the B cell receptor is going to see the antigen. Ig alpha and beta are going to induce the cellular cascade to cause a response. We also have our T cell receptor. We already talked about that. It is an Ig superfamily. It plays a role in antigen specific activation of T cells. A molecule that's related to this T cell receptor called CD3. It's the signal transducing element for T cell receptors. So T cell receptor sees the antigen and the MHC. CD3 is who takes that outside signal and sends it inside to the cell to cause a response. MHC class 1 and class 2 are both Ig superfamilies, and they play roles in presenting antigen to T cell receptors in different contexts. We already talked about poly-Ig receptor. Recall that's what helps get IgA secreted. It also plays a role in, or I guess that was a different one. Poly-Ig receptor helps get that IgA to the outside surface. And then we have a collection of Fc receptors, a few of which we've talked about, like the receptor that helps flip antibody from the mom to the fetus. And Fc receptors will have different specificities based, they will bind a limited range of heavy chains. So you will have Fc receptors, for example, that bind IgG members, and they are called Fc gamma receptors. You might have Fc alpha receptors, which bind IgA. And then a couple of pictures just showing what those look like. These are from your book. You have immunoglobulins. Here we have our Ig alpha beta, so on and so on. And you see a feature for all these are these Ig folds, right? About 110 amino acids that have that are puckered out and form this little globular structure as a result of disulfide chains. And then I included a picture from another text that shows an example of some of our Fc receptors. Should that be interesting? Now, one last kind of like addendum to add is the idea, and this is where I included this because it's honestly cool. There are these things called nanobodies, and what they are opening up is some ideas for new biotechnologies or new therapies. So some animals, and kind of the ones you see most talked about, are sharks, camelids, which include llamas, and those are the ones I'll hark back on. Some animals express these slightly smaller antigen-specific antibodies. So remember, evolution is weird. You get a variety of things. So not everybody's antibodies look the same. Sharks express this form, and camelids can express this type. And what this means is you actually have these smaller structures termed nanobodies that may be better able to bind hidden antigens. And so what's cool is they can take these, they can immunize a llama, for example, and they give it an antigen that they want to develop these antibodies for. They take them, they do some modifications, and what they get are bivalent antigen binding sites that are incredibly small. The reason I bring those up is they have been discussed and are under investigation as therapies for disease. For example, COVID-19, Alzheimer's disease, and cancers. And so they are also robust tools for biomedical or immunological research. You can buy nanobodies to use in your research endeavors. And then I just had this as just a place to look. Why are these things possibly better? They maybe are more heat stable, pH stable, there's less things for your body to see. So they might be less immunotoxic. They might be less antigenic, where you develop a response against them. If you want to see more of that, I have a link out here to follow. And with that, we've finished our antibodies chapter.
The recording has stopped.
Topic, right? And again, this unit is all related to soluble things, but as the antibodies chapter gave good illustration to, that doesn't mean that we will only be talking about soluble immune proteins. Some of these things are also bound to the membrane. And that is very easily observed when you look at cytokines. Now, most of the cytokines we talk about, we are speaking of them as things that get secreted out and to communicate messages from one cell to another. And so we term them as soluble mediators, but really you could think of them a little more broadly as just mostly protein things that communicate in some way. Most cytokines are low molecular weight. They are produced, I have on the slide, innate and adaptive immune cells, but you can find cytokines being produced by things that are not immune in nature. From an immunological standpoint, that kind of makes sense, because if you have, for example, an epithelial cell that's undergoing, it's being damaged, it's being infected, it has the capacity to produce some cytokines to tell everything in the area, I'm doomed, protect yourselves, or bring in reinforcements or whatever. Now, the induced variety, like which cytokines you get, will give you different effector functions. And really, the types of effector functions you'll get will really depend on, one, the environment of the cytokines, but also the cell types who are producing it, as well as the cell types that are responding to it. So what types of receptors are expressed. And a lot of that has to do with the antigen that did the stimulation, right? A viral antigen is going to stimulate a different collection of cytokines than would a bacterial antigen, because they're going to need different responses. And so just a brief word on history. The first cytokine actions were discovered with what was termed lymphocyte activating factor, or LAF. And this was something that was produced by cultured macrophages. And what they found is that they took this thing out of macrophages, and if they purified a bunch of it and put it on top of T cells, those T cells would start to proliferate without any other stimulation. And this is termed, in the absence of antigens, activating things, or mitogens, which are things that stimulate production. So why would a macrophage produce something that would tell a T cell, hey, get going? And that's because, right, we have cells that need to communicate information. Now, early nomenclature, we're going to speak just collectively of cytokines, but we also include a class of molecule called a chemokine. But just to give some context, early before there were a ton of them, and before they realized that many cytokines are produced by a diverse collection of immune cells, they had things called lymphokines, and these were cytokine things that were produced by lymphocytes. And then they had monokines, which were cytokines produced by monocytes or macrophages. And they moved away from that, and they tried to standardize it. And what they did is they did a fairly good job. So if you see the term interleukin or IL, these are things that mediate signals between leukocytes. That is a chemokine or a cytokine. So often you will see described a cytokine that, if you were to look it up, or even in your text, it might have an additional name. And so some cytokines still have two names or more, depending on how it was identified. And many, but not all, will carry with it an IL name. All right, so interleukin-1, which we've already talked about, but you know that we talked about TNF, tumor necrosis factor alpha, which doesn't carry an IL name. So kind of keep that in mind. If you're reading a chapter in the book, or if you're at an immunology trivia when it's safe to do such things, you'll see, if you see IL, think probably a cytokine or a chemokine, something to communicate between two. Now, cytokines in our case are we'll separate them out as a group, but we are talking also about chemokines, and these are proteins that have some sugar modification to them. So they're glycoproteins, and they have chemoattractant properties, which means they cause many different types of leukocytes, especially things like neutrophils, lymphocytes, and monocytes, to move towards them. Right, and so these cells they'll start to interact with IL-8, and then they'll keep moving in a direction that allows them to interact with more and more and more IL-8. And what it's doing, what chemokines are there for, is to recruit cells recruit cells to an area where they're needed, but also to ensure that they're moving in the appropriate way. Now, to take a word about their functions, cytokines are called pleiotropic and redundant, which actually sounds like it could be a very easy contradiction. Like, how can you be pleiotropic, which means widely affecting, but also redundant? And that's important. They are pleiotropic because one cell can produce, we have this activated T helper cell that's producing IL-4. Well, that IL-4 can interact with a variety of different targets. Both immune function is shown here, right? But also non-immune functions. And a good example that's always cool to bring up that your text also talks about, so that it was easy to follow up on. IL-4, especially those that's IL-4 that's derived from activated T cells, contributes to bone synthesis. Or IL-4, cytokines that are derived from T cells has an impact on bones. IL-2 production promotes osteoblastic phenotypes, right? Osteoblasts build bone. And so IL-2 secretion increases the amount of bone that's being constructed. However, TNF actually inhibits that osteoblastic activity. It slows down bone growth. And then IL-4 and IL-13, some other cytokines, actually have differential effects on the behavior of osteoblasts. And all of this is the result of these cytokines that are produced nearly exclusively, or at least in large part, from T cells. And so it's interesting to think during an immune response that could also be having an impact on a completely different system. And the question is, well, why? In the case of T cells and bone, T cells need nutrients, right? And so and the way they get those nutrients might depend on what they're producing. So now, cytokines are pleiotropic. They are also highly linked, and we just have a couple of examples. I didn't use them all from your book, but a couple of examples of how cytokine production is linked to immune dysregulation. This is easily observed in a non-immune setting. Patients that have rheumatoid arthritis, where you have inappropriate activity and targeting of the immune response, they actually will develop osteolytic lesions because that cytokine profile makes the destruction of bone more likely. So you see that as a feature. Now, cytokines are also redundant. Many cytokines will induce similar behavioral responses. And this is explained in part to something we'll talk about in a little more detail later, and that's that many cytokine receptors, so the thing that is on a cell that allows it to see this chemical message, there's a lot of redundancy in those. For example, the receptors that would allow a cell to see ILs 2, 4, 7, 9, and 15 all share one, they have a receptor subunit that's the same. And so what happens is these cytokines tend to induce the same effect or very similar effects. Now, what's the benefit of this? What it means is, one, if you don't have enough IL-4, but you have a bunch of the others, you might still get the effect you want, but it also allows you to mix and match. Effects will depend on the contributions of the different cytokines that are causing different signaling events inside a target cell. Now, cytokines in that way rarely act alone. We may talk about them as being like, an immune cell only responds to IL-4. But that's more on the rare side. Usually a cytokine acts in concert with other things because the environment where all of these immune responses are occurring is complex. So you got lots of things. And what we call a collection or composition of cytokines in a particular area is the cytokine milieu, right? And so this is like the cytokine profile in a particular area. Multiple cytokines can actually influence the actions of others. So beyond just being redundant, you might have cytokines that are additive. The effect of IL-4 and of IL-5 produce, IL-4 causes one response and IL-5 makes it that much more strong. They can also be synergistic, where you have the combination of two cytokines induces a response. If you just give a B cell IL-4, it won't class switch to IgE. If you just give it IL-5, it won't class switch to IgE. But if you give them both, that synergism creates a new response, giving it really a nice mix and match menu that immune cells can do. You also importantly have some that are antagonistic, not only to turn off an immune response, right? A cell that's producing a lot of IL-10 is more likely to turn off immune responses in neighboring cells, but it can also influence it, right? If you have production of IL-4, but a cell starts to produce interferon gamma, that'll actually prevent IgE class switching. And so it's antagonizing the function of another cytokine. And so all of this taken together, it gives you profound amounts of control. Right. Now, functionally, sorry about that. Sorry if your stuff's messed up. Most cytokines have incredibly short half-lives. And so what this means is that the action of a cytokine is typically the result of synthesis, and then its corresponding function will occur in bursts, this oscillatory function, right? Because you need the cell to continuously produce it to get the effect that you're after. Many, I would argue, most of the ones we're going to talk about in the foreseeable future are going to be secreted. So they're going to be soluble, they're going to diffuse out. Some of them, though, can be expressed as membrane-bound cytokines, right? And they, again, communicate between cells in a manner akin to what you would describe for hormones. And so what we can look at is the different effects as a means of distance from target. And so there are some cytokines that are autocrine or can act in an autocrine way. And that is where you have this, say this is a T cell, it starts to produce IL-2. Well, that IL-2 can bind to the receptors that T cell is also expressing, which will promote its activation. They can be paracrine, and this is where you have neighboring cells that are producing cytokines that act on other neighboring cells, so local, like small distances. And then you can have some cytokines that act systemically. We talked about these in chapter 2 when we looked at IL-1, 6, and TNF. If you need the reminder about what they do, those cytokines are the ones that help induce things like fever, right? IL-1, 6, and TNF alpha cause the hypothalamus to change its set point. They also stimulate acute phase proteins, the production of CRP, which then goes and makes the body more inflammatory. Now, how is it then that a cytokine causes an effect if they can be redundant? It's kind of the question of, well, how is it that secreting something into the blood ever hits the thing you want it to hit? Right? Wouldn't it just go and interact with everywhere? Well, that's because target cells will express receptors. And so a cytokine can only cause an effect to a cell. It can only induce a response in a cell that expresses a receptor that can see it. And what that means is that we can change the activity of that target cell, arguably best by manipulating the degree to which it expresses cytokine receptors. Your liver can keep producing, and I'm using an endocrine example, your liver can keep producing glucagon or insulin, but if there are no cells that can respond to it because they don't express the receptors, well, having all of that protein doesn't matter, or all of those hormones, because there's nothing that can see it. And so one of the clearest ways is, yes, changing the amount of cells that are sensitive to a cytokine by changing the receptor expression. You can change it by producing less, right? If you just have less cytokine, there'd be less available. So both of them are meaningful. Now, a good example with changing that cytokine expression or receptor expression, IL-1, which is a very potent pro-inflammatory cytokine, when it hits a cell, it induces the expression of IL-2 receptor. And what that means is that target cell, by virtue of the activity of a previous cytokine, it's more sensitive to IL-2. So this is a good example of synergism, right? IL-2 is only going to act on a cell, in this case, that has been acted upon by IL-1. And so the properties of these cytokines, the types of events that they cause, also shape the immune response. And a good illustration of this is something that we're doing these chapters out of order, so we are introducing the concept of T helper 1 versus T helper 2 responses, but we'll get that in the next unit properly. So use this as, you're going to learn it one way or the other. We're just learning it in a slightly different order. So when you induce cell-mediated responses, they are mostly related to the production of cytokines by activated T cells. One of the interesting parts are these T cells can generate different responses, different cytokine profiles. And one of the easiest ways to characterize these are the Th1 versus Th2. Th1 cytokines, for example, these collection of cytokines here that are going to get produced are the result of a particular antigen that's driving activation and the T cell response towards cell damage and lysis and macrophage activation. Intracellular things are more likely to get going here. You compare that to an antigen that is more external, say it's a bacterial toxin. It will promote a Th2 cytokine response, which you can see these Th2 cytokines here. And what they drive is a response that's more appropriate for toxins, and that's going to be a B cell activation and antibody response. So you can see we're still activating, we still have antigen activating an antigen presenting cell. That antigen presenting cell is going to tell something to a naive T cell, but here the message it conveys is going to be the cytokines, and you have different cytokines that will drive different responses that ultimately lead to the production of other different cytokines that cause your response. Now, another functional property is always nice to revisit chemokines, which again are those chemotactic cytokines. What they do is they recruit immune cells, and in some cases they ensure that an immune cell is going to produce the desired response. You see higher concentrations as you go closer to the secreting source, right? These don't last very long, so as they diffuse away, they not only become more dilute, they also inactivate. So a cell can follow the increasing concentrations to go to the source to do its immune cell function. Cytokines play a key role in helping to regulate expression of cell adhesion molecules, and these are ways that cells can interact with other cells, for the example of extravasating, and that's for a leukocyte to say leave the bloodstream and go into a tissue where it's needed. Cytokines, they're following the cytokine trail that makes that process possible. You can see here we have, I think this is from our previous thing, maybe this is new to this chapter, but we have our steps of extravasation, right? And this is all related to cytokine, or in large part related to cytokines, or at least the effect of cytokines. And so we have this chemokine expressed here, IL-8. There is something going on in this tissue that's stimulating production of IL-8. And that IL-8 causes these neutrophils to start to pay attention, right? So they start to roll because they're now interacting with the endothelial cells. And what they are doing is responding to the IL-8, and the IL-8 is making this process much more likely to leave the bloodstream and enter into the tissue to respond. Now you have common systemic functions, so beyond the events locally, leaving the bloodstream, activating a cell, you also have systemic effects, which in large part are the amplification of an immune signal. It's like taking one cell who's producing some IL-4. Well, that IL-4 can go and interact with 10, it might be producing enough cytokine to interact with 10 other cells, and then those 10 cells can amplify and interact with now 100. And so you see that a small degree can grow into a big one. And a good example of this amplification process that cytokines can perform is the idea in delayed type hypersensitivity, the TB skin test, or contact dermatitis like poison ivy. You have very few of these self-reactive cells that respond. Let's say you had 10 T cells that were able to see this obscure antigen that it shouldn't be responding to. But what happens is those 10, because of them getting activated and producing cytokines, you now have tons of monocytes being drawn to the area in numbers that far exceed the number of originally activated T cells. And so these systemic effects, right, this amplification, just the activity of cytokines often can be beneficial. But it can drive diseases, which we'll see. And there are some ways to exploit these cytokine dysregulations therapeutically. I think I've just kept one example from your text at the end. Now, cells produce different cytokines. They also respond to different cytokines. And what this can lead into is the idea of a cytokine cascade. And these are the interactions of multiple cytokines during a typically progressing immune response. The type and the exact cascade that gets stimulated is really a large determinant of the type of adaptive immune response you're going to get. Is it going to be Th1 versus Th2 versus Th17 versus B cell activation? And this is a result because the antigenic stimulus promotes the cascade that will lead to a particular type of cytokines that are going to be produced. And of the cells that do this best, really, it's your helper T cells. One of their best function is to produce a particular type of cytokine profile that drives particular immune responses. Now, these cytokine cascades, not only driving a response in a particular direction, they also help control or contribute to how long this activity is going to take place. This is really through the balance of pro-response or anti-response, so stimulating cytokines versus inhibiting cytokines. They can also influence other systems. So you could have a cytokine response towards an antigen, but it could be influencing behavior of the brain or neurons or whatever. And again, these cytokine cascades can be good where they're inducing protection and immune response, or they could be bad. And the good example of this is the cytokine storm that is a feature of allergic reactions, infectious diseases. Now, bad side, right? We're in the middle of the SARS-CoV-2 pandemic. You can click on this link and that'll take you to a paper that describes the cytokine influences of it, which may be interesting, but this is an interesting way to look at not only how a cytokine storm might get generated, but also how that then ultimately affects other tissues, other organ systems. So SARS-CoV-2 infects epithelial cells. They influence, this is NLRP3 is our inflammasome from chapter 2. They stimulate activity, and what happens is they infect and end up triggering the production of large amounts of cytokines that are activated that are secreted from macrophages, which themselves stimulate the activity of lots of T cells and macrophages and neutrophils. And what you get is tons of cytokines being produced, especially those that are related to pro-inflammation. And this creates the cytokine storm. So many cells are being activated and secreting cytokines at once that it starts to overwhelm all of the regulatory processes. And this can ultimately lead to organ dysfunctions. Microcirculations can lead to hypoxia or impaired neurological function. So yeah, interesting and concerning. And that's where you get the idea that one thing that might be used to treat some of these diseases is to target, not necessarily try to get rid of the pathogen, but also try to control the inappropriate immune responses that might result. Cytokines can be broken up into functional categories. And so we're going to talk about the major cytokines that are of relevance to our course. There are many, many that are there. but you will see a motif of the ones that keep popping up. IL-1 is a good example. And what we're going to do for our course, we could classify these in a variety of ways. I figured I like the text does this in a way that works for our class, and that is to group them together by their function, not necessarily by their structure or receptor or whatever. And so a note to make sure we're clear, many of these cytokines are pleiotropic. And so what we're going to do is we're going to focus on the major functions or the functions that are the most interest for the purposes of our discussion, class, lecture, etc. Now, a good place to review these is table 12.1. The images I was trying to grab of it, it's such a big table, it's a whole page. It looked bad on the slide. So I'd refer you to table 12.1 as you're organizing and reviewing cytokines. It's a good place for a quick recall. So what cytokines facilitate innate immunity? Principally, I put these in two groups because we can think of them in two responses, maybe. We have IL-1, which if you see IL-1 alpha and beta, yes, they are different, but they are together IL-1 cytokines. IL-6, and then TNF alpha. And we've already spoken of these in the innate immune system chapter. And we talked about these in the context of that pro-inflammatory response. We'll see them again in a few slides. And what they do is principally these get produced when there is a pathogen that's stimulating them. They produce these, and these, in this case, are going to activate innate immune cells. We also have the activity of interferon alpha and beta. So first, our IL-1, IL-6, and TNF alpha. Largely, it helps coordinate an early response to an infection. And largely, they're produced by phagocytes. So a phagocyte would eat something, it would see it with its pattern recognition receptors and say, oh, crap, this is bad. Let's start producing IL-1 and IL-6 and whatever. Now, the biological consequences of the production of this suite of cytokines is you're going to elevate body temperature, right? These three cytokine types are collectively called endogenous pyrogens because they cause the hypothalamus to reset to be warmer for reasons that we've already discovered that you'll want to make sure you remember. But things get better and bacteria grow worse. Our IL-1, 6, and TNF alpha also increase the number of neutrophils that are present in the bone marrow. They increase the activity of antigen-presenting cells so that they are better able to inform the adaptive immune response. And they stimulate the acute phase response, which is the response that causes liver cells to produce things like CRP, C-reactive protein, and one we'll talk about in the next chapter, mannose binding lectin, or MBL. What's interesting about both of these is or about all acute phase proteins collectively is they make things, they boost the generalized immune response that ultimately is to try to get rid of something as fast as possible. The two that I've highlighted, CRP and MBL, both activate complement, which will serve us well as a segue into our next chapter. Just a reminder, what are the results of complement activation? You could get lysis, you could get opsonization, where you make something more attractive to eat, and definitely you'll get inflammation. And this is again in the context of an immune system related complement activation. Now, I separated out interferons alpha and beta. These are type 1 interferons, and what these type 1 interferons do is they help minimize the spread of viral pathogens. Many different cell types can produce interferons, and typically they start to produce them once they are infected with a virus. The most potent of these are plasmacytoid dendritic cells. These were first identified as cells called natural interferon producing cells because they produce so much of it. It is true that many non-immune cells can also express interferons. Epithelial and endothelial cells are good examples. Now, you're trying to minimize viral spread. So there's kind of two ways to do this. You can produce this to make your neighboring cells more resistant to infection. You can also more resistant to infection and to help withstand the antiviral responses that are on their way. But you can also make yourself the interferon can also make you a more attractive target for elimination. Like, something's wrong with me, I'm producing interferon, you need to take care of me. So when you look, interferons, the type 1 interferons are really good at enhancing MHC class 1 expression on cells that are not yet infected. And this is kind of the withstanding the consequence of activation of the antiviral response. As natural killer cells start to come in, recall natural killer cells are, their default position is to kill unless they get that typically MHC1 handshake where they say, oh, you're good, so I won't kill you. So by making sure uninfected cells are expressing good amounts of MHC1, they're not going to be accidentally killed by the natural killer cells. Interferons can also enhance the MHC1 expression on infected cells. And a reminder, all cells that have a nucleus express MHC class 1. What this gives them a unique ability to do is not only do they express MHC class 1, but MHC class 1 is where you can put antigens in it and they can get expressed. And so by causing infected cells to express more MHC class 1, the natural killer cells and activated CD8 T cells, which we'll talk about in chapters something in 8 or 9. These cells are going around looking for cells that are expressing MHC1 proteins that activate them. And so what this can do is by causing more infected cells to express MHC1, you get more death of these virally infected cells. Now you're going to have a collection of cytokines that help regulate the adaptive immune response. And as you might expect, this regulation is going to relate to altering the behavior of B and T cells as a result that follows antigen stimulation. And you're going to get these different responses based on the cytokine profile that's induced. Some cytokine profiles will promote or inhibit proliferation, activation, differentiation, as well as controlling the duration and intensity of the responses. And now, since in the adaptive immune response, we need antigen-specific responses, how do you ensure that only antigen-specific lymphocytes respond to these cytokines? And there's cool, I split them up into 3 ways. One, only cells that need to be activated are going to express the functional cytokine receptors. Only T cells, and we're going to use T cells as an example, only a T cell that is in the process or that has been activated will express the appropriate cytokine receptor. And this is what you see here. This T cell is getting activated by the appropriate signals from an antigen presenting cell. And in doing so, it starts to express the right cytokine receptors that allow it to continue its activation. Another thing that helps ensure you only have, that you only have activity of the cytokine function on the right places is you have the fact that co-stimulation, right, the ability to respond to these cytokines requires cell-to-cell contact. You can see this very clearly here. And actually, this is exaggerated. Probably for future chapters, I'll use the more appropriate image. But what happens is you form this immunological synapse, very akin to what you describe with neurons, where the 2 cells have this space where events are occurring. That intimate contact ensures that there's less chance that your secreted cytokines are going to the wrong place. And this type of interaction is called a cognate interaction. So the cytokines produced as a result of this cognate interaction are most likely going to affect the nearest cell, which happens to be the one that's physically contacted. The other thing, which is just a general scheme of cytokines themselves, they have a short half-life. And so there's less chance for the cytokine to leave an area where you want the activation because they don't last very long. That reduces the likelihood of those cytokines going and activating the wrong group of T cells or adaptive cells. Kind of in line with an adaptive immune response, lots will be discussed about how T cells get differentiated. Those are very good examples of the differential effects that cytokines have. And so just another word about Th1 versus Th2, and here you can call this skewing, differentiation, polarization. And what we mean is we're taking a naive T cell, it's going to see antigen, it's going to get the right cytokines, and then it's going to shift to one of a number of tracks based on which cytokines are being delivered. And so cytokines are the key determining step in how a naive T cell that's been activated, and here you will see this called a Th0, which subset, which types of T cell response is it going to move down? And so in the IL-12 and Th1 response, so it turns out IL-12 is a key cytokine to cause a T cell response to go towards your Th1 response, which recall is best for dealing with intracellular pathogens, macrophage activation. So a antigen presenting cell is going to activate a T cell and it's going to produce IL-12. IL-12 is especially common in barrier tissues that are the antigen presenting cells from barrier tissues. And what IL-12 does in this naive T cell is it increases the amount of Tbet that's being produced, and Tbet is a transcription factor which leads to the production and secretion of a specific group of cytokines. So here you'd say IL-12 is related to T helper 1. So this is the polarizing cytokine that causes a specific transcription factor to be produced, which leads to the production of these specific cytokines. And it's interferon gamma, GM-CSF, TNF alpha, IL-3, and TNF beta. All of these, this profile will make the environment and will lead that T cell to activate things that will deal best with intracellular pathogens. You compare that to the T helper 2 response. And here we're looking at IL-4. So an antigen presenting cell or a cell that's been stimulated, say a mast cell that's seen a parasitic antigen, it's going to produce lots of IL-4 in the process of that T cell being activated. What will happen is that will lead to a specific transcription factor termed GATA3. So again, a lot of immunology is knowing who goes where. And one of the places you see, if you see Tbet, you should think first T helper 1. If you see GATA3, think first T helper 2. Then you can start to add the layers on top. And so if a naive T cell has GATA3 being produced, it will produce IL-4, 5, 10, IL-3, and TGF beta. This collection of cytokines will cause this T cell, or the T cell that is producing this collection of cytokines will enhance the production of antibody in the neighboring cells. And this is important because here we're dealing with parasites, extracellular parasites or toxins. These are threats that are best dealt with antibodies. What happens if T helper 1, which is intracellular stuff, viruses, intracellular bacteria, and T helper 2, which are parasitic or external toxins? What about like bacteria and fungi that are extracellular that don't fall neatly into this? And this is through the discovery of a T helper 17 subset. So Th17 cells were identified because IL-17 family cytokines were discovered. And they are, there's a table in your text that has the different ILs, but IL-17s, but simply it's IL-17A, B, C, D, E, and F. Most of them, especially IL-17A, most of them are very pro-inflammatory. What they do, and one of the key places, but not only, they're produced by a type of a differentiated T cell called a T helper 17. And what they do is they promote robust inflammation. Th17 cells are good at getting neutrophils to come and causing cell death and destruction. And so they play a good role in responding to extracellular pathogens, but unfortunately, they also have a significant influence in certain types of autoimmune and inflammatory disorders, especially things like asthma and inflammatory bowel disease. Now, how do we get to that Th17 response? Here we have the combined activity of IL-6 and TGF beta. If a cell is secreting IL-6 and TGF beta, an activated T cell is that T helper 0 is going to be skewed towards the Th17, and what that will cause is the production of within that cell it's ROR gamma T, and what this gets said is ROR gamma T. The transcription factor ROR gamma T leads to significant production of IL-17 family cytokines, which again drive neutrophil proliferation, increase neutrophil activity, events that are best to deal with extracellular bacteria. Now, this is a snapshot that you'll see again when we talk about T cells, and this is our T cell subtype polarization paradigm. Here we have an activated T cell. We could call this T helper 0. It's interacting with this antigen, and then this antigen on an antigen presenting cell or something that it can see antigen on. And what will happen is, depending on the cytokines that get produced as a result of this interaction and as a result of the environment where this is occurring, you could get one of many different pathways. You could get Treg cells, or you could get T helper 2 or T helper 1. And so this, although we're not talking about all of these in context yet, this is a good place to identify, to kind of streamline the events that are occurring. And you might see some differences. So if you, for example, this is talking about IL-12 and IL-8, you'll default to the ones that we talked about on the previous slides, because we're keeping our naming consistent. It's important to note too, we have these T cells driving, in the case of a T helper 1 versus T helper 2, those cytokines that are produced during a T helper 1 response will also impair T cells from going to a T helper 2 response. It's kind of like, don't do two things at half effort, do one thing at very strong effort. So most of what you see, for example, is the cytokines that get produced in a T helper 1 response actually impair the ability of a T helper 2 response, and vice versa. And I borrowed this from another text where T-bet, the transcription factor for T helper 1, actually interferes with transcription factor GATA3. And so in a T cell that's being skewed towards T helper 1, that's going to turn off its ability to be skewed towards T helper 2, right? So it ensures that a T cell is going to go towards one of these trains. What's interesting is that if you take T helper 0 cells and you just give them TGF beta, no IL-6, they actually become, or most likely to become, a type of cell called a T regulatory cell. And these are very inflammatory. And so there's a pre-built network to not only make active T cells, but also to make some that are inhibitory. All right, and so we have another functional class, and these are going to be those that are pro-inflammatory. We are seeing some that we've already seen before, IL-1 and IL-6 and TNF alpha. These are pro-inflammatory cytokines. We're just now looking at them in the context of global function, right? We talked about their innate activity before, but innate activity leads to inflammation too. And so what do these pro-inflammatory cytokines do? And I have drawn here interferon gamma, which is especially important by being produced by macrophages and acting upon macrophages to chew up, to become more phagocytic. And so the biological functions would be to help increase vascular permeability. Here, that gives access to the immune cells. It aids, it works and acts in concert with chemokines that are being produced. And that not only gives you more access, but it ensures that they are coming to the right location, as well as activating and enhancing the function of inflammatory cells, which ties back from before. Things like neutrophils are stimulated and are more active in the presence of these pro-inflammatory cytokines. Now, the functions of these pro-inflammatory cytokines are limited. One, cytokines just don't have a long half-life, but two, as a response goes, you'll start to produce what are called regulatory cytokines. And this is to help control the immune response so that it does not run away and create like a cytokine storm. And so an example of some of these regulatory cytokines are TGF beta and IL-12, right? And here you see IL-12 is not only listed in the pro-inflammatory cytokine, but also as a regulatory. And that's because it depends on what a cell is expressing as to how it'll behave. So lots of gray areas. So I borrowed this from the text really as a means to show how some of these pro-inflammatory responses can work, and again, in conjunction with cytokines. And I've just simplified this. In panel A, what we are looking at is we have here, this is the endothelial cell, and this is our neutrophil. And here we have E-selectin and ICAM. These are molecules that will allow for a neutrophil to bind, to slow down, to try to to do something. In the absence of any cytokine signaling, no IL-8, which is a chemokine, in the absence of that, we have weak interaction between E-selectin and the specific neutrophil ligand. what happens is you start to see more of these inflammatory cytokines, and what they do on the endothelial cell in this case is they cause them to produce lots more E-selectin. And what that will allow is more interaction between the neutrophil and the endothelial cell. And if you couple that together with the fact that in this case, there's now lots of IL-8 being produced, that IL-8 will draw the neutrophil towards it, because IL-8 on the neutrophil will increase the specific integrins, these glycoproteins that are expressed that interact with things like ICAM1. So what you have is weak interaction until you get the pro-inflammatory cytokines, plus the chemokine allows strong interaction so that that neutrophil can move into that tissue. Ooh, should have cleaned that up, huh? So a word about chemokines. How do cytokines as a functional group affect leukocyte migration? Well, again, chemokines are closely related to their low molecular weight glycoproteins with varying things. They are potent attractors for many different leukocytes. Some chemokines have additional roles in things like respiratory burst, which is how macrophages and neutrophils try to kill. The release of particular enzymes, calcium mobilization, things like angiogenesis. And if you think about it, all of these make sense because the chemokines are trying to recruit things there. Well, what better way to recruit things there by also giving them a boost towards their activity? Chemokines can be complicated because you'll start to see lots of weird or lots of unusual groupings. And so what I've done is just group these in a simple way to show that one, chemokines are organized into 4 subfamilies, and what they are based on is the structural motifs that occurred at the amino end of the protein of the sequence. That's fine. Instead, you could also just learn what the subfamilies name are, and then for a few, I have characteristics, chemokines of note. So you have a subfamily of chemokines called the CXC, and just so you know, X is a variable amino acid. It could be anything. So, and the C's are cysteines. So we have CXC subfamily. The principal one here that we'll focus on is IL-8. CXC family as a whole are widely produced. They play big roles in inflammation and repair. And they are very good at recruiting neutrophils, but also driving their activation. And so that's why we see IL-8 here. You then have the CC subfamily, and important ones here are RANTES, MIPS1 alpha, and MIP1 beta. These are pro-inflammatory. They collectively contribute to Th1 responses. And what's interesting about these is that they bind particular receptors. And so this is a cytokine receptor called CC, which means CC subfamily receptor 5. And what's interesting about that is that that's one type of HIV enters your cells through CCR5. And so by one way that this could work is if you have increased production of these cytokines, you can actually outcompete the HIV. They don't have places to go, and that could slow down infection. And then you have 2 other subfamilies that we might come across as we move through the rest of the chapter, but it's not worth bringing up anybody yet. You have the C subfamily, and you have the CX3C family. Now, another functional class are those cytokines that stimulate hematopoiesis, so the production of more blood cells. Collectively, these cytokines can be called colony stimulating factors, or CSFs. And there are several distinct pathways. There could be overlap, but we'll kind of treat them as independent as possible. You have those that are the macrophage CSFs, or MCSF. And as the name suggests, these are going to cause clonal expansion, so the production of more macrophages and monocytes. You have GSF, which will cause the production of granulocytes, especially neutrophils, because they represent the biggest proportion of those produced. You then have GM-CSF, granulocyte macrophage CSF, and these stimulate the production of macrophages, monocytes, and granulocytes. You have IL-3. IL-3 is a stimulator of a variety of blood cells, especially in the presence of erythropoietin, which you probably talked about in anatomy as the thing that controls red blood cell production, which is produced from the kidneys because the kidney has lots of blood flow, so it knows the state of oxygen carriage. And then the last is IL-7, so that we don't forget our adaptive immune cells. IL-7 induces the differentiation of lymphoid stem cells into B or T cell progenitors. So IL-7 is what helps shift that bone marrow production, gives you more of those adaptive lymphocytes. And so those are our functional classes. And so we've already mentioned, oh, how does a cytokine act only on one cell? It does so because that target cell will express the appropriate receptors. Now, receptors are arguably the thing that's very highly regulated. You know, not only is a cell producing, but once it's producing, if you look at a resting cell, very likely it's going to express no receptor, or it expresses low or intermediate affinity receptors, which means the binding is not going to be terribly good, which reduces the chance that cytokine will activate that cell. And a good example of this is IL-2 receptor expression on resting T cells. If you look at resting T cells, they express a dimer of IL-2 receptor called gamma beta. And they stick this out, and gamma beta is not great at binding IL-2. But once that T cell starts to undergo activation and stimulation, it expresses an additional alpha chain. And what happens now is that the alpha, gamma, and beta chains form a trimer. And that alpha subunit, when together with the beta and gamma chains, is high affinity. So now you have the ability of that T cell to interact or to respond to IL-2 stimulus, solely because when that T cell was undergoing activation, it's expressed the chain that allowed it to see best cytokine to promote its proliferation and continued activation. So if you're looking, and so both of these examples could be considered the resting state, but we'll focus on the middle one. In the resting state, a T cell will express gamma and beta. It binds IL-2 okay, but not enough to cause robust activation. If you were to just express alpha alone, it binds IL-2 very poorly. But in this case, at the end here, this is a high affinity IL-2 receptor because these three chains are expressed together. And so here you see the affinity. So the lower the number here, the more strongly they're interacting. So this ensures that a cell that is intended to respond to IL-2 will respond to IL-2. Now, the receptor families are divided into subgroups, and I have them split up over 2 slides. So you have our Ig superfamily, and these are the ones, recall that they have possible. I don't know why possible. They have Ig fold domains. Some of these receptors will respond to things like interleukin-1 and MCSF. You have class 2 cytokine receptors, and typically class 2 receptors have 2 chains. The alpha chain is usually the one that does the binding, and when it's together with a signal transducing chain, which is either a gamma or a beta, that creates the most affinitive relationship so that you get the best response. There are some exceptions. The previous slide, we talked about IL-2, and that receptor forms a trimer. Most of the cytokines that have been discussed or identified are going to bind through class 1 cytokine receptors. Class 2 cytokine receptors are also known as interferon receptors, and since that is their name, got lots of typos here, sorry about that. And since that is their name, they interact with, their ligands are interferons of varying degrees, and the types of receptors you express will give you your different responses to different interferons. The fourth subfamily, or the fourth family of cytokine receptors, is our TNF receptor superfamily. This class is very big. It gets divided up into 3 groups: those that are death receptors, those that are decoy, and those that are activating. All of the subgroups, whichever one of those 3 they fall in, all have similar extracellular binding domains. So they see TNF in a particular, they're all fairly similar in the way they see TNF, but then they have different capabilities to transduce that signal. And this is mediated through TNF receptor associated factors, or TRAFs. And these are adapter proteins that allow particular responses to occur. Now, what's interesting about the TNF receptor superfamily is that their ligands can be those that are membrane bound. And I've gone ahead and pulled out, you can have TNF receptors called FAST. Fast activation or fast signaling is what drives predominantly apoptosis in an immune context. And then you also have CD40 is an example of a TNF receptor member, and its interaction helps drive B cell activation, but you also have ligands that are soluble, kind of the traditional discussion. You also have one last group, which are chemokine receptors. And chemokine receptors, throwing back to cell biology, are considered multi-pass transmembrane proteins, which means they work through, they pass through the membrane, I think 7 times. And as a part of that, they are G protein coupled receptors, which means cleavage of G protein is what transduces the signal to go from outside to move it through in the inside. Now, some of these chemokine receptors also play a role in allowing pathogens to enter. We already mentioned HIV. Some forms of that can bind CCR5, and in binding that, it allows that HIV to enter through, and that's a chemokine receptor that it's doing that with. And here you're looking, again, your Ig superfamily with their characteristic Ig fold domains. We have our class 1s and our class 2s, and they are different based on the conserved sequences they have. We have our TNF receptors, and then our transmembrane spanning chemokine receptors. What's nice about this slide is it gives you a little illustration about who the ligands are. You can see class 1 carries with it the bulk of the cytokines that we would have come across with. Now, I've already mentioned you have that idea of the redundancy, right? Cytokines can induce similar. One of the ways to do this is they all share this common chain, or many of them share a common chain. And specifically, it's the common gamma chain. This is most often, and it's for many cytokine receptors, it's the signaling transducer, right? And so it's what's going to allow the outside message to be conveyed and passed through into the cell. And receptors that use this gamma chain include the IL-2 that we already talked about, but the IL-2 receptor, the receptor for IL-4, 7, 9, 15, and they all share that. Now, although they'll have this common gamma chain, they'll have a different chain, typically alpha or something else, that will be who binds to the unique cytokine, or type 1 cytokine. We can see those here where our green chain is going to be the gamma chain, which is the consistent signaling one, and then different combinations of the others that give you the ability to see different cytokines. A brief word about signal transduction. Cytokines will induce intracellular events, and what that will result in is the production of a transcription factor, which leads to a particular group of genes to be expressed, which give you the biological effect. So in most class 1 or class 2 receptors, these are the simplified events to focus on. Cytokine binding on the membrane is going to induce the cytokine receptors to cluster together, and this will cause either dimerization or polymerization, depending on the type of receptor. And what that will do is it'll bring, and hopefully this is familiar from cell biology, but it will cause the cytoplasmic tails to get close together. And what that will do is it will allow them to activate each other. And upon doing that, it allows a type of kinase called a JAK kinase to come in there, and it allows them to build or to have these docking sites so that they can ultimately result in the activation of STATs. And our STATs are transcription factors that once they are activated, they'll dimerize, go into the nucleus, and cause our gene expression. This is what we're looking at. On the normal cell, you will just have some of these receptors just sticking out. And then once you bind one, it causes them to dimerize. And when that happens, the cytoplasmic tails can now hold on to JAK. JAK can activate STAT. When you get STAT activated, and it can dimerize, it'll dimerize, go into the nucleus and produce, say, more cytokines, or produce the ability to migrate to a particular location. Now, how is that cytokine get turned off? There's a discovery of a collection of signaling terminators called suppressor of cytokine signaling, or SOX proteins. And what these do is they target specific proteins that are associating, and in targeting them, they cause them to degrade the signal. And think of this as as a cell is responding to a cytokine, these SOX proteins are going to accumulate, and it's akin to negative feedback. And I've borrowed this from a review from the literature. This is the normal event that occurs, and so we have the cytokine, we get our JAKs, then our stats. That stat's going to go into the nucleus, and it's going to cause the production of gene. But as those genes are being produced, you're going to slowly accumulate the amount of these SOX proteins you produce. And as that production goes up, it's ultimately going to go in and turn off cytokine signaling. This is important to control the amount of cytokine activation that happens, because we'll see in the next slide or so, cytokine storms, this rampant production of cytokines that are not controlled or that last for too long, are always detrimental to the organ. Even if it's getting rid of the pathogen, it will damage, destroy, or severely influence normal activity. All right, and so just 2 brief slides, really, because we're going to hopefully talk, time permitting, we're going to talk about more. We're going to talk about more diseases in context, in groupings, and later in the textbook. And so just to give that flavoring now, I pulled out 2 from the text of cytokines that contribute to disease. Overexpression or underexpression of either the cytokines or the receptors can promote disease pathologies. And so 2 good examples, which are related somewhat to the cytokine storm aspect, are toxic shock syndrome. And toxic shock syndrome is a disease, or is a pathology initiated by toxins produced by certain species of Staphylococcus. It's, at least I remember it best from microbial or pathogenic microbiology in the context of like a tampon that's left in too long. And eventually these staph organisms will have the chance to grow. They'll start to produce these toxins that accumulates. And the problem here is that these toxins are a class of molecule called superantigens. And we'll talk about this when we go into T cells, but to introduce it now, a superantigen is an antigen that is so good at binding T cells, it doesn't have to have a specific antigen. What superantigens do is bind the T cell receptor and bind an MHC, and it forces the T cell to activate. And so you get direct activation, and superantigens are really good at causing lots of IL-1 and TNF alpha production, giving you your systemic effects of the disease. The other is bacterial septic shocks, or sepsis. And here, sepsis is most associated with the endotoxin, which is a specific part of bacteria, a specific part of the cell wall of gram-negative bacteria. And what LPS, or endotoxin, does quite well is it induces macrophages to produce large amounts of IL-1 and TNF, and this leads to excessive immune system activation, which leads to the multi-organ dysregulation and susceptibility to other, because all your cytokines are being produced inappropriately. Now, just a brief word, you can exploit this, and I've just pulled out one to look at. How could you exploit these for disease? There are lots of monoclonals that target, TNFs and different receptors and all that. But to be a little more simple, a brief example are the idea of cytokine inhibitors or antagonists. And as their name implies, their purpose is to prevent cytokine function, either to neutralize the soluble cytokine or to prevent the receptor from working. And a good example are soluble TNF receptor molecules, and these have use in treating rheumatoid arthritis. And what they do is that when they are placed, instead of having your cytokine that can interact with the receptor, instead, the cytokine is going to interact with these, call them decoy receptors, or these soluble receptors that don't stick to anything. So there's no way to cause activation. And what this effectively does is it reduces the amount of TNF that could activate the cell, hopefully reducing excessive or inappropriate inflammation. I think that's it. So the crash course of cytokines is over. I'm grateful for you all being able to move to Zoom today. Anybody have any questions? Let's start recording.
The recording has stopped.
Cool. All right.